So today we are joined by Tyler Cuddy, who is completing his PhD at Cardiff University. And Tyler will be presenting findings from his PhD project, which investigates heavy metal bioaccumulation in Bornean carnivore within forests surrounding palm oil plantations. So without any further delay, I'll hand over to today's speaker.
Thank you very much, Betty. Hello, everyone. My name's Tyler Cuddy. I am a PhD student. I've just gone into my third year at Cardiff University, and I'm with the Eco-Risk Centre of Doctoral Training. And I am part of the Organisms and Environment Division within Cardiff University. So today, I am here to talk about my PhD project, which is looking at tropical Mesopredator ecology and the effects of heavy metals on the environment. This project is in partnership with JNCC as part funders, and I've actually just completed my 3-month secondment with the environmental pollution team within JNCC, and it is also in partnership with UKCEH and Danau Garang Field Centre, which is where my fieldwork is based. So to give you a little bit of an overview with the project, where I work and where I'm posted on fieldwork, I was out there for 7.5 months last year, is an area called the Lower Kinabatangan Wildlife Sanctuary. This is situated within northeast Malaysian Borneo, and it is a series of 10 fragmented forest lots which have been retained along the Kinabatangan River, which is very much the river that's the lifeblood for that region. This area was from sort of the 1970s, 1980s onwards. It fell afoul to the timber industry, which got rid of many of the large fruiting trees, the dipterocarp trees, and those were all systematically logged out. And later on down the line, there was then the surge of the oil palm industry, which has resulted in many of these forest lots that have been retained being surrounded on all other fronts by the oil palm industry, which is very, very prolific in Southeast Asia and along many equatorial regions. So, with all that in mind, habitat fragmentation is a primary threat for many of the species within the region. However, despite that, some species have shown very, very high levels of adaptability to that agricultural matrix, be that for getting between forest patches or for resource acquisition, which is quite often the case. And some species have even started to move into the plantations and live a large proportion of their time within those plantations because there is plentiful resources. That's all well and good that the populations are doing well and have adapted to this change. However, this persistence does bring them into more regular contact with agri-products. And there's a lot of extensive fertilizer use within those plantations. And many of these chemicals that are used have components that are potentially harmful and have invisible impacts on the wider community. These can come from fertilizers, rodenticides, herbicides, fungicides. Another one is palm oil mill effluent, which is a type of wastewater that comes from the oil palm extraction process. And whilst in that area there is regulations as to how much of this can be released into waterways at a certain time, it is not very well enforced. It is not policed. So you do have quite a few instances where there's a surge of that being released into waterways. So this project aims to assess how heavy metals from some of these sources may impact the carnivore community, and if there's any species that we can use there that are a bit higher up the food chain that we can use as potential bioindicators. So, primary objectives of this project is A, to assess the heavy metal levels in various different biological tissues from some focal species, which I will introduce in full a little bit later on. And I also plan to assess the heavy metal contamination levels in potential prey items and try to relate dietary composition to heavy metal loads to try to trace what the primary vectors for these possible contaminants up the food chain are. So for the dietary analysis, that will be the main genetics component of this project alongside the ecotox side of things. And we will be using dietary metabarcoding of fecal samples taken from trapped individuals and environmentally sourced samples, and investigating the contaminant levels in those, and then relating dietary composition to those contaminant loads for an individual. And then lastly, to understand how heavy metals from agri-products are transported through the local food chain and the potential later implications for carnivore communities. So for a little bit of context for the oil palm and Borneo specifically, Borneo, the whole island, is considered one of the 4 main biodiversity hotspots of Southeast Asia, and there is a high level of endemism on the island. But the island has lost the vast majority of its natural forests, especially the primary forests. Much of the forest that remains is now secondary, so lacking those large dipterocarp trees. And the oil palm sector, it contributes hugely to the Malaysian economy. It encompasses 5% of the total Malaysian agricultural exports and produces 41% of global vegetable oil. Borneo as a whole, so between Malaysian and Indonesian Borneo, produce approximately 85% of the world's palm oil. So it is very prolific on the island. And Sabah alone, despite being quite a small province, produced approximately 5 million tons of oil palm in 2020, which was 6% of the entire global production. The reason that the oil palm industry is so prolific is to do with its productivity and its how productive it is in equatorial regions. For every 5 metric tons of palm oil extracted, there's about 15 million tons of fruit bunches produced per hectare in a year. And then the plant itself, Elaeus guinensis, it fruits all year round. So it's got that constant productivity that can be relied upon to be able to export a product all year round. And that product itself is hugely versatile. So it's used in pizzas, vegetable oils, it's used in soaps, some cosmetic things. So it is very, very high demand within multiple different industries. Obviously, this increased demand has led to rapid expansion, and that has fueled a lot of habitat loss. The issue with this, in the West, the oil palm industry gets somewhat demonized. But when you compare it to what the next best alternative is, you would need to clear about 50% more forest to get anywhere near the yield to meet the demand with the next best alternative. So it's the presence of oil palm is kind of a best case scenario, and because it is so ingrained into the socioeconomic development of so many equatorial and developing countries, the trade itself isn't going to go away. But it's about finding that coexistence and trying to do that process sustainable. That is the focus, rather than trying to make the exports stop for it and the development of that farming practice stop in that, because it's just not going to happen. But down at the bottom, there is a little bit of indication as to how much was put into the environment chemicals-wise in 2020 across Malaysia. So you've got 36,000 metric tons of pesticides, 386,000 metric tons of nitrogen, 172,000 of rock phosphates, 1,050,000 tons of potash, and that is just in Malaysia. That's not taken into account Indonesia as well for the island. So very, very extensive. So why are heavy metals the focus of this project? So heavy metals are present as trace elements within many fertilizers, and fertilizers are used extensively within the oil palm plantations, the largest states. The thing with heavy metals is that they are a legacy chemical. They are very, very environmentally persistent because they cannot be degraded or broken down. They can be biotransformed into various different sister products, but those are quite often more detrimental than the actual element itself. So an example of that would be mercury. Mercury is, in a biological system, harmful in very small amounts, but one of its products that it can be broken down into is methyl mercury, and that is much worse than standard mercury. So the issue that we find in that general area is that these heavy metals can leach into the surrounding environment from the plantation runoff, and a lot of that is down to the fertilizer application regime. Where we work is a floodplain, and as I mentioned, it's right next to a large river as well, which regularly breaks its banks. So a lot of the fertilizer that gets applied in that region is applied topsoil. And of course, if there's a flooding instance, that is all just going to be lifted and taken elsewhere. And that is usually out into the surrounding forest. There's quite a few different types of fertilizers that are used within that region. And that can have different levels of trace elements within them. And heavy metals themselves are generally split into two categories. So you've got your essential heavy metals and your non-essentials. Essentials are used in biological processes, but for biological systems, there's kind of a Goldilocks zone for each of those essentials. So if there's too much, it's detrimental to the system. If there's too little, detrimental to the system, but that can shift from species, taxa. It can even shift within the same species. So male and female might have different sort of optimum levels for an essential heavy metal. Age can influence it, reproductive status. So there's a lot that influences that optimum range. The non-essential heavy metals, however, are not used in any biological processes and are very harmful to a system in very small amounts. And they're especially a severe threat for animals higher up a trophic scale, so higher up the food system, because of a process called bioaccumulation and biomagnification. So if you've got, say, 5 rats that have been feeding around fertilizer or a rodenticide, they are each themselves taking up those chemicals. But then if you've got a wild cat, or I'll be introducing you to civets if you don't know what those are as well, that comes along and eats all 5 of those rats, it's going to be accumulating everything that was in each of those rats. So it magnifies up the food chain. The targeted heavy metals for this study are the essentials: copper, iron, zinc, cobalt, magnesium, manganese, nickel, and chromium. And then the non-essentials are cadmium, aluminium, lead, arsenic, and mercury. So I'm now going to introduce you to the main focal species of this project, even though some other species do crop up in the data set. But yeah, you'll get to know a bit about the various furry things and not so furry things that I chase in the jungle that have made it abundantly clear that they don't want to be caught. But first of that roster is the Malay civet, or Viverra tangolunga. This is one of the largest of the Bornean viverids. Viveridae are the family that have civets and genets in them. And these guys can get up to about 7 kilos. They're more terrestrial hunters. They don't tend to use any of the canopy, and they are highly generalist. They've got a lot of protein within their diet, but where that protein comes from, they're not really fussy. It's usually small mammals. They go for invertebrates a lot as well. And they'll go for lizards, and they'll also eat various fruits and berries that they find around the forest floor. This species specifically has been found from a previous study to persist within the plantation matrix. Albeit not very deep, they'll only tend to move about 200 meters or so into the plantation, but they'll move in overnight because they are nocturnal, and then move back to the forest for the rest of the day for them to find their sleeping sites. So they're more of an edge specialist, and they are also the species that my project kind of had the foundations laid for by a previous PhD student here at Cardiff University and with Dan Algarang Field Centre as well, who did the initial ecotoxicological assessment of this species and found the individuals which she had radiocollared as well, that were using the oil palm habitat quite a lot, had higher levels. If I go back a couple of slides here to this box plot, showed higher levels of cadmium, aluminium, lead, and interestingly, lower levels of mercury than individuals who just stuck to the forest. That kind of paved the way for the development of my project. Now we're taking a multi-species approach to build on the knowledge that she developed. Another species that I'm looking at, these cute little guys, the common palm civet, Paradoxurus hermaphroditus, very, very widely spread across Southeast Asia. And they are a lot smaller and more arboreal, up to about 4 kilos, and they tend to make use of the canopy, and they are largely frugivorous. So yes, they are also very generalist in their diet, but they have a preference towards fruit, which is nice, having some generalist species that are closely related but have different preferences within that generalist niche. But they have also been found to regularly utilize the plantations due to easy resource access. Unlike the Malay civet, who get to sizes that are a bit too large to be able to find suitable sleeping sites within the plantation and then move back to the forest for the day, these guys have been found to make use of the plantations for sleeping sites as well. They'll be able to make use of what little understory vegetation there is. They're also able to make use of the canopy for sleeping sites. So they tend to persist a bit deeper into the plantations. And then the last of the mammalian carnivores, which I'm focusing on, is the Sunda leopard cat, Prionailurus bengalensis javanensis, bit of a mouthful. And they are arguably the most ubiquitous species within plantations when it comes to mammalian carnivores, or so the hypothesis is. Again, they are very small. You're talking only about 2.5 kilos. And they have the capacity to be able to persist deep into the plantation matrix because they can make use of that little understory vegetation, and they'll also make use of that canopy for the oil palm themselves. And they are, as with all cats generally, obligate carnivores. So yes, they are generalist, but they stick to protein for their diet. And the oil palm industry loses approximately $6 billion of yield each year globally to rat and mouse damage. So plantations are very much a hotspot for rodent activity, which has, in quite a few cases, enticed leopard cats in because it is good hunting grounds for them. And that entices them to stick around. There's somebody who I am collaborating with at Dan Algarang Field Centre to get samples from Sunda leopard cat called Amanda Wilson. And she is working on collaring them and understanding more of their spatial use and spatial ecology within that sort of fragmented landscape. And there's been individuals that have spent 95% of their time upwards within the plantation. Females that have bred, had their kittens within the plantation. Their kittens have grown up somewhat and then found their own space. So they persist a lot. There's one individual in my data set who is a kitten who's found about 1 kilometer into the plantation. I haven't analyzed them yet, but very interested to see if they're showing any signs of contamination levels, because if they are, when they're only a few months old, then that has huge implications for the future of this species moving into the plantations more and more. And the fact that they are used as what's referred to as an IPM species, which is an integrated pest management species. So essentially a form of biocontrol for usage within sustainable oil palm plantations, much like pine martens have been introduced as biocontrol in certain areas in Scotland for grey squirrels. And then onto the big lizards. So this is the Asian water monitor lizard, so Varanus salvator, and they are very common in that area within the plantations and the forests, and they are what you'd refer to as a hypergeneralist. They are not fussy at all about where their food comes from, whether it's alive, dead, long dead, they don't care. But they persist within the plantations very, very efficiently as well, much akin to why the mammalian carnivores are there, plenty of resources. And also what entices them in is the amount of water waste that there are within a plantation. An oil palm estate is very much reticulated by waterways because and drainage ditches, because each tree can go through about 120 liters of water a day. So they're very, very water intensive, which is why you also get extensive runoff into the surrounding waterways, because any fertilizer that is washed down into these drainage ditches, that leads out to the tributaries that then feed into the main river, and so on. Hopefully we'll get to understand a little bit more about the fate of those chemicals as well. These guys are also, they're cannibalistic sometimes. And when they get to larger sizes, they can get up to about 50 kilos. They will eat smaller monitor lizards as well, and many of the mammalian carnivores that I am also focusing on because they're a lot smaller. And there's literature that suggests that scavengers are of potential higher risk of bioaccumulating harmful toxins because there is no selectivity for the food items that they're going for. If a leopard cat comes across some rats that have died of poisoning, or for any reason really, they're going to pass that up. They're going to go for the ones that are still alive and kicking. But these guys will come along, and it doesn't matter how the other animal has died. They're going to count that as an easy snack. So to give you a bit more context as to the area that I'm working in, so the orange star there signifies where Danau Garang is, where we are stationed in relation to the sample sites. And then there's two larger states that are rather close by. There's Hilco, which is on the opposite side of the river, and about 50 meters to 200 meters away from the riverbank. And then you've got another plantation called Penderosa, which is on the same side as the river, but a bit further along. In the Lower Kinnan Patangan Wildlife Sanctuary, there's a rule that's governed by the Sabah Wildlife Department that a plantation has to maintain a boundary, a riparian buffer between the river and the edge of the plantation of at least 20 meters. That varies in certain places, but it means that there is along the river, for the most part, a continuous green strip. But it doesn't take long to get from the river to these estates. Hilco and Penderosa are both quite sizable. They're all about 2,000 hectares. And Hilco itself is certified by RSPO, which is the Roundtable on Sustainable Palm Oil, who are a certification for sustainability practices. So Hilco and Penderosa both have what are called HCV areas, which are high conservation value areas, which are sections of forest that are retained within the plantation boundary to keep a bit of animal traffic and give animals the capacity to be able to get from forest patch to forest patch without having to actually go into the plantation. Penderosa itself is not currently certified, but Hilco and Penderosa are both owned by the same overarching umbrella corporation called Felder Global Adventures, not Adventures, Felder Global Ventures. And you would think, because they are owned by the same overarching company, that they would both have the same general processes with how they do things in the plantation, but that is not the case. Hilco itself have NPK, ERP, borate, and phosphate fertilizers applied about 4 to 5 times a year. And the amount of fertilizers that are applied is based off of a third-party agronomist assessment, which happens twice a year. And that infers what quantities to put out the fertilizers. And Hilco use commercially available fertilizers, which we will be able to get our hands on, hopefully, and analyze ourselves as well to see exactly what levels of trace elements you're seeing in those. But Penderosa, however, they apply irrespective of what the what an agronomist says, about 4 to 4.5 kilograms per tree per application, and their applications are a lot more regular throughout the year. And obviously, when you've got 135 trees per plot, and a plot is a hectare, and there's 2,100 hectares, that is a lot. I haven't done that maths, but that is a lot of fertilizer going into that area. And this is based on the recommendation of a Felder-specific agronomist. And what's really interesting as well is that they apply their own fertilizer that is not commercially available. So we don't really know how much trace elements and stuff like that are within that fertilizer, which makes it a little bit trickier to deal with. And whether or not we're going to be able to get our hands on any, don't know. But we'll have to wait and see on that front. So, as I mentioned, last year I was out in the field for about 7.5 months. And over that time, through battling with floods and so on, we were able to get a decent roster of data. So you'll see that there's otters and another type of cat there as well. So I try to be as generalist and opportunistic as the species that I study, and make use of all the samples that we find environmentally as well. So we did stumble across some otter sets. So hopefully I'll be able to look at the dietary composition and the heavy metal levels in fecal samples of otters within the plantation and within the along the river and within the forest, as an assessment of aquatic hunting carnivore, and then be able to compare that to the terrestrial side of things. And then the other feline there is the flat-headed cat, which is actually a semi-aquatic cat that's only about sort of 1 to 1.5 kilos, very, very small, but critically endangered. And we do happen to have a couple of samples from them. But they are extremely, extremely elusive. Hopefully, I might be able to get some more samples from them, but they're very elusive and very hard to catch. So we caught 10 individuals of Malay civets, 15 monitor lizards, 6 common palm civets, currently 7 leopard cats. And then when it came to the potential prey items, we were doing a lot of small mammal capturing as well. So we caught 74 different individuals there, and then looking at that aquatic food sources as well within the various different waterways, we caught approximately, I think it was 201 fish all in all. So to go into the methodology for how we get these samples, for the carnivores and for the monitor lizards, we use active trapping. So there's 2 different types of traps that we use. What you can see in the pictures there, and what I'm currently climbing into to stuff a bait box full of rotting chicken, which was a lovely way to start many of my days. That's what's referred to as a gravity trap. So you put the bait at the back, and it's got a cradle system which pulls a wire that is connected to a pin or a pole that is holding the door aloft. And then when somebody steps on the trapdoor, pulls the pin out, and then closes the door, and gravity does the rest of the work, hence the name. For the monitor lizards, they generally are more active during the day. So we would do diurnal trapping for them. And that would involve going out in the morning, baiting with rotting chicken generally. And then each of these traps were monitored by a camera trap as well. So we could have a look at what sort of activity we were seeing around the traps. Those had their SD cards changed once every couple of days. So we were keeping pretty regular watch of what was going on around the traps. So they'd be set in the morning, and then checked sort of mid-afternoon past what are the main activity times for monitor lizards. But when we were trapping for the mammalian carnivores, we would be doing nocturnal trapping. So they'd then be reset in the evening and then checked early morning. For the civets, we would bait. It took a little bit of refining, but we combined 3 different types of bait for them. So a bit of palm fruit at the back of the trap as a visual lure, because it's very brightly colored. And then we would have an audio lure, which is this little battery-powered squeaker, which sounds like a ground-nesting bird of sorts, and that would be in a box strapped to the back of the cage. And then there would be a scent lure, which was used cooking oil, which would be laid in trails then from the door of the trap along various different like mammal trails and movement trails that you could see around the trap, and then checked early morning. And if we had an animal in the trap, that would go to sampling. So for the mammalian carnivores, so that 1, we can minimize risk to the animal, and 2, minimize risk to ourselves, they would be anesthetized. This project was done working alongside a very experienced wildlife vet, Dr. Reza Tamizi, who you can see there tickling the underside of a monitor lizard. Just seems to be the best way to calm them down. I don't know whether it's because they like it or it just confuses them to no end, but it settles them down pretty regularly. But for the mammalian carnivores, they would be anesthetized, and once they were fully under, we would take their biometrics. We would take a fur sample, take a blood sample, and then take fecal samples if possible. And then for the monitor lizards, because they're reptiles, they have much slower metabolisms, anesthetizing them isn't really possible in the field. So it would be a case of approaching the head with a large towel. This would be Rez's job. I'd be on the other side of the cage holding the tail, so that he could grab a hold of the head with enough time to momentarily pin it down, and then we put a zip tie on the nose so that no, it couldn't bite us, and they would calm down pretty quickly in the hand, especially after a tummy rub. For larger individuals, their back legs might need to be bound, but none of it harms the animal. All of it was done under the correct processes and with ethics approval from Sabah Wildlife Department, and obviously all the right permits were in place for this. And then the perks of not having to anesthetize them, once the process is done, we can just take off the zip ties and then send them straight off, and they take off at a heck of a lick. So I mentioned that we take fur samples from the mammalian carnivores. We take scale samples as an equivalent from the monitor lizards. So those would be usually done in the form of tail notch, because they have this large sort of dermal ridge along the length of their tail. And this is soft, sort of fatty tissue with a lot of scale tissue there as well. The reason for taking these various different sample types is because we are looking to use them as proxies for different lengths of exposure to various different elements. So for hair and scales, we're looking at the long-term exposure. A hair, for instance, the follicle is in constant contact with the blood supply. So it is constantly metabolizing and uptaking elements such as heavy metals, because they bind very, very readily to the to various aspects of the hair follicle, and they're taken up into the hair. But once they are in that keratin matrix, they're kind of locked in place, because it's such a stable matrix. And with that, you get an idea of long-term exposure, chronic exposure to a certain element or toxicant, which is why hair is used in drugs testing and stuff like that, in sports and so on. And then the blood samples is a proxy for what is in the system at the time of capture. So that more sort of short-term time frame. And then the fecal heavy metals analysis will be as a proxy of what is moving through the system. So then we can kind of look at what uptake rates may well be, and how much of what is going through the system is actually bioavailable to the species that it's in. And here's a few release videos. You've got a Malay civet on the top left there, doddering off quite slowly. I think she was still a little bit woozy, bless her. And then a palm civet that tried to take off a little bit too quickly, and as you can see, the monitor lizards do not hang around. What's nice is that civets are actually rather trap happy. Once they know that there's a snack in the trap, they will quite often come back. They're not the brightest sparks, but they're adorable, so it makes up for it. But there was one individual, because of course everyone that was caught has a name, and there was one individual, Eddie, who decided to go back into the trap for, I believe it was 8 nights in a row. He clearly wanted the snacks, and he also made it his mission to break my things as much as possible, one night after another, more and more as the time went on. So you do kind of get to recognize them, and that some of them have quite a personality. And with Malay civets especially, you can sort of start to recognize them from a distance, because the marking on the side of their necks is essentially their fingerprint. So that's always nice. So looking at the heavy metals in the potential prey items, you are looking at a fairly similar process to same sort of through process as what we're doing with the mammalian carnivores and the monitor lizards. Capture, take scale samples and fur samples, and then that will be used later on down the line for the heavy metal analysis. Alongside the mesocarnivore and mesopredator trapping, within 20 meters of each of those trapping sites, there was a small mammal trap. So we would be doing that in tandem, and then fishing in a lot of the waterways using a combination of gill netting and cast netting. And then these samples will later down the line be used for heavy metals, doing a process called inductively coupled plasma optical emission spectroscopy, ICP-OES. So essentially for the heavy metals lab work, the samples get digested, well, they get cleaned and then they get digested in nitric acid. And then this is loaded into the ICP-OES, which is a, albeit less sensitive version of mass spectrometry, but it's much cheaper, and you can do a lot, you can bulk load it with a lot more samples. As we all know, I'm sure sometimes the ecological sector can be not as well funded as others. So being able to look at these ecotoxicological assays and be able to investigate these sorts of things for a fraction of the cost could be very beneficial to the field moving forwards. And then for the dietary analysis, the fecal samples are prepared using what's called the two-step method, where they're doused in ethanol and then dried out. And then DNA will be extracted from these, and we will be doing metabarcoding, which will allow us to use various different types of genetic primers to select for what's called the CO1 gene. And this is selected due to its mitochondrial lineage. Therefore, it has a very fast mutation rate, and it is present within all eukaryotic creatures. And this will allow us to identify closely related species within the fecal samples, hopefully. And there's just a little bit more in detail about the protocol when it comes to the ICP-OES. As I mentioned, every sample is digested in acid. Then they are diluted using millicube water and filtered, and then loaded into the ICP-OES, where it is volatilized, so turned into a fine mist under high pressure, and that feeds up into a argon plasma flame. And depending on what the wavelength of light is emitted by the flame when the sample goes into it, a couple of sensors can then translate that into an output for a certain calibrated chemical in milligrams per kilogram. So we haven't been able to do much of the analysis yet. As I mentioned, I was on my secondment for the last 3 months. And we also had some large delays in getting the samples actually back to the UK. But this is just a very, very quick representation of what we're seeing for the mammalian mesocarnivores. And just a few trends to highlight, one in specific. So down on the bottom one here, we've got the Malay civet, common palm civet, and leopard cat arsenic. In the bottom here, as you can see, leopard cat, extremely high levels of arsenic within its blood. Blood is the only sample that I have the data generated for so far. Hopefully over the next coming weeks, I will be able to generate the rest of the data, and I've included my email account in this presentation. So if you want to know more and you want to get in touch, please do. But that's just a quick visual representation of where we're at with the analysis. Haven't done any rigorous stats yet, but that is all to come. So to summarize, this project is aiming to investigate heavy metal contamination levels amongst persistent generalist mesopredators across the plantation management schemes and the forest within the Lower Kinabatangan Wildlife Sanctuary. We want to trace down the primary modes of contamination, relate dietary niche and dietary composition to those heavy metal loads. And this will be done using blood, hair, and fecal samples to assess the health of the carnivores overall. And this will allow us to have a deeper understanding of how intensive agriculturalization is affecting the surrounding environment using what will hopefully be suitable mammalian and reptilian sentinel species. And of course, I couldn't do it all without all these lovely people. So a massive shout out to the team at Danau Garang Field Centre and also everyone that I work with through JNCC and Cardiff University and UKCEH. Thank you very much for listening, and any questions?
Thank you very much, Tyler, for that presentation. Really enjoyed it. I'm sure everyone else did too. We don't have any. Oh, we do have a few questions popping up in the Jamboard now, which is great. Cool. I do have one of my own, actually, which I'm going to go with first, if that's okay.
Of course.
I was wondering for your methods and future results, if there's any consideration for maybe more the mammal species of females offloading heavy metals onto young in the womb, and if you think that might potentially affect differences in males and females of population as well.
So this is hopefully something that I can do a bit more work with, but it is very dependent on what goes into our traps. We can only work with what decides to grace us with its presence, and the effort to success with catching carnivores isn't as high as I would like it to be. But I did mention the kitten that we had in the data set, which was picked up by plantation workers about 1 kilometer deep into a plantation. So that is a clear sign that that individual was born within, and its den was probably nearby. As I said earlier on, there's been cases where females have given birth and raised their young within that plantation matrix. And hopefully, because we weren't able to get all 3 different types of sample from them, they were too little to take blood from. But age-wise, they couldn't have been more than a few months. They'd probably only been on hard food for maybe 3-4 months, hard food for no more than a few weeks, judging by the size of it. So we've got fur samples and we've got fecal samples from them. And if they are showing high levels of, say, arsenic, like the other leopard cats are showing in their blood at the minute, then that is potentially sign of female offloading onto the offspring. And there is potentially large implications then for that species moving forwards, because them raising their young within that matrix isn't, it seems, a rare thing, or at least so the hypothesis says. So there is the potential for looking into that, but whether or not it will be more than just a coincidental, here's this data point and what we found, but whether or not we'll actually be able to do any rigorous stats off of it and have more than one instance of that in the data set is yet to be seen.
Yeah, so thank you for answering that. I imagine it's quite that's a whole different topic, maybe. Yeah. So you have a few other questions. Are there plans to disseminate the findings of key stakeholders? So, for example, with farmers?
Absolutely. So something that we'd really like to be able to do with this project is be able to present the thesis and the data to plantation managers, plantation owners, and also Sabah Wildlife Department. One as an indication as to what the wider impact of the industry is in that area, but also one of the key things with trying to get any policy changed in these sorts of areas, or see any development on that side of things, is to give a economic benefit to the plantation managers and the companies that run them. So for instance, if we can show that there's high levels of contaminants being found in the forest that's related to fertilizer application, fertilizer use, that is clear indication that all of the fertilizer that they are applying is not going where they want it to be. So that could be an incentive moving forwards to either apply your fertilizer in a more beneficial way, or B, use less fertilizer to save yourself money, but if you apply it this way, you'll see the yields that you're seeing anyway. So it's just a case of trying to frame the project with that later down the line, when disseminating it to stakeholders, giving them that economic benefit and incentive, which we'll hopefully be able to support with the outcome from this project. Because at no point do we want it to be pointing the finger at the industry, because as I mentioned at the start of the presentation, it's kind of the best of the bunch. It's the best alternative, but it is about finding that coexistence with it and trying to do things in the most sustainable ways moving forwards. And if you can give an economic benefit to the workers and the owners to do so, then that's the way forward, I think.
Thank you. And next question is, what's the purpose of having a visual scent and the other lures in the trapping process for your methods?
It was basically just a whittling down of process and a bit of a pilot phase where we were sort of just tracking success of the trapping when we used one type of bait and then another type of bait and then those two baits together. And it just transpired that as we were trialing things, having that triple threat really helped with the success of those individuals. I must say, we never got a cat in any of the traps. They're caught in a very different way, and that's again in collaboration with Amanda Wilson, who I mentioned before, who's doing her PhD at Danau Garang and is focusing on the leopard cat as her focal species and is doing a lot of spatial ecology and radio collaring of them. Best way to get them is to go out in a truck in a plantation at night with good head torches, look for the eye shine, and then jump off the truck, attempt to surround the cat all with big nets and hope you can net it. And that is the best tried method that seems to have the best success rate, because they are just too smart to go into one of the traps. But thankfully, the civets aren't the brightest sparks. But especially having that audio lure really helps with enticing them in.
Thank you. I find it quite funny as well in your methods when you were talking about after trapping the monitor lizards, monitoring the
Yep.
Final question for the moment. What do you think is the main reason for the difference in the approach to fertilizer application between the two palm oil sites, given they were owned by the same corporation?
Not really sure, to be honest, because they are so. Penderosa is a little bit lower down in the floodplain. It's got a bit less elevation to it than Hilco does, and it weirdly enough it has a bit more understory vegetation, it seems. But they are a lot more rigorous with their chemical application. So they use a lot more fertilizer, they use a lot more rodenticides, they seem to use a lot more pesticides. So that, by all assumptions, will be a bit of a hotspot for various different chemicals going into the environment, or so we expect. Unfortunately, sampling it in that region last field season wasn't really doable for much of the field season because it did spend a lot of time underwater. Hence why I say it's got a bit of a lower elevation than Hilco does. But we haven't quite been able to make sense of it yet. It's purely, it seems, down to manager preference. So there are some things that are kind of governed somewhat by the overarching company, but how a manager runs their estate is not one of those things. So it could just be the manager of one and the manager of the other just have very different opinions on how things should be done. And that's the best answer I can really give for that, I'm afraid.
Brilliant, thank you. I think that is all the questions that we have so far. Just double check there's none hidden. Yeah, I think that's all of them.
Okay, great.
Brilliant. Well, thank you very much. That was a
Thank you
really,
very
really
much.
interesting
It's been a
talk.
pleasure.
Yeah, and I hope everyone has a lovely day. Thank you.
Thank you very much, everyone.
Bye.
Thank you, Tyler.
Cheers. Bye-bye.
Tyler, that was great.
Thank
Thanks.
you.
Thank
Bye.
you. Cheers. Ta-da.